In this module, we will introduce concepts relevant to electrochemical cells. We will start our lecture with some foundational definitions. Oxidation is a process involving the removal of electrons from a species, while reduction refers to the addition of electrons. Together, oxidation and reduction are integral parts of a redox reaction, short for reduction-oxidation reaction. A redox reaction is a chemical process in which oxidation and reduction occurs simultaneously. It is characterized by the transfer of electrons between different species and leads to changes in the oxidation states of the reacting species. In redox reaction, the reducing agent or reductant donates electrons or acts as an electron donor. By doing so, the reductant undergoes oxidation. The oxidizing agent or oxidant accepts electrons or acts as an electron acceptor and reduces. Redox reactions are often accompanied by the movement of atoms or ions. To illustrate the definitions of oxidation and reduction, let's consider the reaction between copper ions in an aqueous solution and metallic zinc. An atom of metallic zinc loses 2 electrons and forms a zinc ion. It acts as a reductant, donates electrons, and oxidizes. A copper ion gains 2 electrons and forms an atom of metallic copper. It acts as an oxidant, accepts electrons, and reduces. Any redox reaction can be broken down into 2 separate half reactions. A half reaction is either the oxidation or reduction component of a redox reaction. It is obtained by considering the change in oxidation states of individual substances involved in the redox reaction. These half reactions are theoretical constructs that individually represent the transfer of electrons. This approach is not limited to typical redox reactions. Many reactions that don't initially appear to involve redox processes can also be expressed as 2 oxidation and reduction half reactions. For instance, in the example of the reaction between zinc and copper ions, the half reactions are as follows. In the first half reaction, representing the oxidation, the zinc atom loses 2 electrons to form a zinc ion. In the second half reaction, exemplifying the reduction, the copper ion gains 2 electrons to form a copper atom. The overall redox reaction is the sum of these 2 half reactions. Now we will introduce the concept of a redox couple. A redox couple consists of the oxidized and reduced forms of a species involved in a half reaction. We denote a redox couple as OX and red, and its corresponding reduction half reaction is written as shown here. In this equation, N represents the number of electrons transferred in the half reaction. It is important to note that the standard way of writing half reactions is always in the direction of reduction. Therefore, oxidation can be theoretically viewed as a reduction process occurring in the reverse direction. Continuing with our example involving copper and zinc, the half reactions for the redox couples are as follows. For the copper redox couple, The half reaction is copper 2 plus 2 electrons produces a copper atom. In the example redox reaction considered before, this half reaction runs in the forward direction. For the zinc redox couple, the half reaction is zinc 2 plus 2 electrons produces a zinc atom. In the example redox reaction, this half reaction runs in the reverse direction. Previously discussed redox reactions can occur spontaneously, for example in solution, by transferring electrons upon direct contact between reactants. But now we will explore how these reactions can be controlled and utilized within an electrochemical cell. An electrochemical cell is a device that spatially separates the oxidation and reduction processes that constitute the overall redox reaction. Similar to a redox reaction being separable into two half reactions, an electrochemical cell can be separated into two half cells. A half cell consists of an electron conductor, typically a metal, being in contact with an ionic conductor, typically immersed in a solution containing ions that can undergo a redox reaction. This interface between two phases with different types of conductivity, where electrochemical processes take place, is the most significant part of the half cell and a focal point of electrochemistry. Importantly, each half cell in an electrochemical cell must contain species that constitute a redox couple. In a complete electrochemical cell, two half cells are connected, allowing for electron transfer between them through an external circuit. If there is an actual redox reaction taking place in the electrochemical cell, the electrode where oxidation occurs is referred to as the anode, while the other electrode where reduction takes place is called the cathode. As the reaction progresses, electrons released during the oxidation at the anode travel through an external circuit before re-entering the cell at the cathode, where they drive the reduction process. To illustrate this concept, let's examine the electrochemical cell depicted in this figure. This cell is a classic example of an electrochemical setup consisting of two half cells. On the left, a zinc electrode is immersed in a solution of zinc sulfate. The solute dissociates into zinc ions and sulfate ions. On the right, a copper electrode is immersed in a solution of copper sulfate, which dissociates into copper ions and sulfate ions. These two half cells are connected by a salt bridge, typically a gel containing an electrolyte such as potassium chloride. The salt bridge facilitates ionic current flow between the half cells while preventing the direct mixing of the solutions. A metal wire attached to each electrode completes the external circuit, allowing for the flow of electron current. This example demonstrates the fundamental setup of an electrochemical cell, where the physical separation of oxidation and reduction sites enables controlled electron transfer, essential for various applications in electrochemistry. The overall chemical reaction that occurs in an electrochemical cell when the two half cells are connected is referred to as a cell reaction. This reaction encompasses both the oxidation process happening at the anode and the reduction process occurring at the cathode. The cell reaction represents the sum of the individual half reactions from each half cell and illustrates the transfer of electrons and the transformation of reactants into products within the electrochemical cell. In our example, the cell reaction is the reaction between copper ions and zinc that we had started from. When this cell reaction is allowed to proceed spontaneously, zinc at the anode undergoes oxidation, releasing electrons and zinc ions into the solution. These electrons travel through the external circuit to the copper cathode. At the cathode, copper ions in the solution accepts these electrons and are reduced to copper metal, which deposits on the electrode. This flow of electrons from the zinc anode to the copper cathode generates an electric current, while the chemical changes at each electrode demonstrate the underlying redox processes driving the cell. The thermodynamic equilibrium in an electrochemical cell is closely connected to the concept of cell potential or its electromotive force. An electrochemical cell reaches equilibrium when there is equilibrium at every interface and the electric potential difference at the cell's terminals is compensated by an external power supply. The latter means that there is no current flowing in the cell. This electric potential difference, E, that counteracts the thermodynamic driving force of the cell reaction in equilibrium, is called the electromotive force, EMF, of the cell or cell potential. If there is no equilibrium at every interface, the measured potential difference is not the true EMF. The relationship between the cell's electromotive force and the Gibbs energy of the overall cell reaction bridges the gap between electrochemical and chemical thermodynamics. When an electrochemical reaction is carried out reversibly, the electrical work done is the product of the charge transferred and the potential difference through which it moves. This reversible work, when performed at constant pressure, is also equivalent to the reaction's Gibbs energy. From this understanding, we derive a fundamental equation that connects cell potentials to thermodynamic properties. Delta Rg of the cell reaction equals the negative of N times F times E. In this equation, N represents the number of electrons transferred in the cell reaction. F is the Faraday constant and E is the electromotive force, EMF, of the cell. The EMF of the cell is defined to be positive for spontaneous cell reactions. This is highlighted by the negative sign in the equation. A positive EMF corresponds to a negative delta Rg, indicating a spontaneous reaction. Using an experimental setup like the one previously shown, we can measure the cell's electromotive force, which represents the difference in electric potential between two half cells. This measurement inherently includes contributions from both half reactions occurring in the cell. Importantly, such measurements do not yield the absolute electric potential of each individual half cell. To effectively discuss and analyze electrochemical reactions, we need to know the potentials of individual half cells. For specific reactions, controlling the potential difference at the interface is crucial. It is also not feasible to tabulate standard cell EMF values for all possible combinations of redox couples. In addressing the challenge of measuring half-cell potentials, a practical approach is adopted in electrochemistry. We select one particular half-cell to serve as a reference, to which we assign an arbitrary electric potential of 0. This reference half-cell becomes a standard against which the potential of all other half-cells is measured. By combining a half-cell of interest with the reference half-cell, the potential difference measured across the full cell can be attributed to the analyzed half-cell. By dividing the electrochemical cell into two separate half cells and splitting the overall cell reaction into its constituent redox half reactions, we can then characterize each half cell by its electromotive force when it is connected to the reference half cell. The EMF of the full cell is then determined as the difference of potentials for the two half cells. The standard hydrogen electrode SHE plays a fundamental role in electrochemistry, serving as the reference half cell with 0 potential. This electrode is essential for defining the electric potentials of half cells. This figure shows a schematic drawing of the standard hydrogen electrode setup. It comprises a platinized platinum electrode immersed in an acidic solution and exposed to hydrogen gas. To maintain standard conditions, the activity of hydrogen ions must be 1, which is equivalent to pH equal 0, and the pressure of hydrogen gas must be 1 bar. The half reaction of the standard hydrogen electrode is 2 hydrogen ions plus 2 electrons in equilibrium with a hydrogen 2 gas molecule. The electric potential of the standard hydrogen electrode is defined by the thermodynamic equilibrium between hydrogen ions and hydrogen gas. The standard hydrogen electrode is the primary reference for the electrode potential scale. Its electrode potential is set to 0 at every temperature. This standardization allows for the comparison of the potentials of other half cells when connected to the standard hydrogen electrode. By using the SHE as a reference, we can determine the potential of individual redox couple, which is crucial for controlling interfacial potential differences in specific reactions and for understanding the electrochemical behavior of various systems. Electrode potential is defined as the electromotive force of a cell formed by connecting the half cell in question to the standard hydrogen electrode. This definition implies that the electrode potential is not an intrinsic property of the half cell alone, but includes a constant contribution from the interfacial potential difference of the standard hydrogen electrode. The significance of setting the electrode potential of the standard hydrogen electrode to zero is that it allows us to assign electric potential values to individual half cells by measuring their electromotive force relative to the standard hydrogen electrode. This figure provides a conceptual illustration of the process leading to the determination of electrode potential. It shows a cell consisting of a half cell and the standard hydrogen electrode. In such a setup, the entire electromotive force of the cell is attributed to the half cell under investigation. For example, if we measure the cell potential of a zinc half cell against the SHE, the measured electromotive force of the entire cell is assigned as the electrode potential of the zinc half cell specifically. Let us consider a half cell containing a half reaction represented as oxidized species plus n electrons are in equilibrium with reduced species. Besides the chemical species undergoing actual oxidation or reduction, other chemical substances can participate in the redox half reaction. The electrode potential of a half cell or of the respective redox couple that it contains is called the standard electrode potential if all reaction participants are in their standard states. The Nernst equation allows calculating the electrode potential of a half cell or of the respective redox couple when it is not in the standard state. It is given by E equals E standard minus R times T over N times F multiplied by the natural logarithm of Q. Here, N is the number of electrons transferred in the redox half reaction, and Q is the reaction quotient. The reaction quotient Q is the ratio of the activities of the products to the reactants, each raised to the power of their stoichiometric coefficients. Note that the activity of the electrons does not appear in Q. The Nernst equation is useful in determining how changes in concentration and pressure of reaction participants as well as of temperature affect the electrode potential of a half cell. The electrode potential of a half cell can be also expressed by the equation E equals the negative of delta RG divided by NF. Here, delta RG represents the reaction Gibbs energy for a theoretical reaction where the half reaction in question is balanced by the half reaction of the standard hydrogen electrode. This equation indicates that the electrode potential is an intensive property. It remains unchanged even if the stoichiometric coefficients in the reaction are multiplied by any integer, as both delta RG and N change by the same factor. How does the cell potential relate to the potential of individual half cells? To determine the overall cell potential, we combine the electrode potentials of the two half cells making the electrochemical cell. This is done using the equation E cell equals E red minus E ox. In this equation, E red represents the electrode potential of the half cell where reduction occurs, and E ox is the electrode potential of the half cell where oxidation occurs. The electrode potential of the oxidation half cell is taken as negative because the electrode potential is defined conventionally for the reduction direction. This approach allows us to calculate the electromotive force of an electrochemical cell by considering the individual contributions of each half cell.